Hello, I'm Dr. Marie McNeely, and I'm excited to welcome you to episode 662 of the show. This week, we are resharing our interview with Dr. Olaf Andersen, originally published on June 1st of 2015. Listeners, Olaf has done extensive research on cell membranes and membrane proteins to better understand how small molecules can enter and impact our individual cells and our bodies as a whole. His work has a lot of really important applications for the fields of pharmacology and toxicology. So everyone, get ready to learn more about Olaf and his work in this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am thrilled to be here today with Dr. Olaf Andersen. So welcome to our show, Olaf. How are you?
I'm doing very well.
Wonderful to hear. Well, we are excited to chat with you today. Now, listeners, a little bit about Olaf. He is a professor of physiology and biophysics at the Weill Medical College of Cornell University, as well as director of the Tri-Institutional MD-PhD program in New York City. He was awarded his MD from the University of Copenhagen in Denmark and completed postdoctoral research there, as well as at Rockefeller University, before joining the faculty at Cornell University. Now, Olaf has received many awards and honors during his career, including being named a foreign member of the Royal Danish Academy of Sciences and Letters. He has received the K.S. Cole Medal from the Biophysical Society. He has been named an Honorary Fellow of the Cornell University Weill Medical College Alumni Association. He has received the Distinguished Service Award from the Biophysical Society and has also received the Inaugural Ballard Mentoring Award. And now Olaf is here with us today to tell us about his journey through life and science. So Olaf, today we want to get to know you, both as a scientist, but also as a person. So can you start off by telling us what you like to do when you're not doing science?
When I'm not doing science, I read a fair amount. But I think the two things I really have fun doing is I'm an avid home brewer, and I really get a kick out of brewing beer. And my ambition as a brewer is to make a beer that has a really deep, full taste with very little alcohol.
Interesting.
So I was out in Salt Lake City when the Biophysical Society was there some 9 years ago, and I was amazed by what the brewers in Utah could do. They can't brew anything that's stronger than 3.8% in alcohol. And most people who brew beer of that alcohol strength make something that is pretty close to water. It could be lawnmower beer, but it's really not worth it.
Gotcha.
And that was excellent, quite frankly. Any idiot, if you're allowed to go to 8 to 10% alcohol, it's hard not to make a beer that people will think tastes well.
Gotcha. You can't mess it up.
If you stay below 5%, and particularly if you begin to go, I've not been able to go below 4%, but if you get to that, then you actually now have to begin to think. You have to really find out, how do you get the mouthfeel, how do you get the test, how do you get everything to work out in such a way that you are proud of serving your beer and people actually line up to get it. Because one of my recruitment ploys as program director is that at our annual holiday party, which we have just after we accepted the applicants into the program for the next year, I serve my home brew, and that's only going to be a recruitment ploy if the students think that it is a reasonable product. So I have a lot of fun out of that.
Absolutely. Well, this sounds like a fantastic and even somewhat scientific endeavor. I like it.
Yeah, I have a 60-page brewing diary. I've been doing it for a little more than 20 years, and I can tell you how every single batch I have been making over the years has been, with notes, what works, what does not work, and so on. It's fun.
I like it. You've got your Beer Lab notebook. So what are you known for?
Well, I think the best I'm known for is actually my IPAs, which I make, as I said before, not that strong, but where I particularly explore using oat chips in the beer itself. So many times IPAs are characterized just by being very strong, very hoppy. that certainly is a characteristic of most IPAs. But years ago, when I read about the origin of the style, it occurred to me that why not explore what happens if you put a little bit of oak chips into the beer, and it tasted better. You really can taste it. And then one time, I just for the fun of it, I normally used American oak. I saw that the guy whereby my homebrewing supplies, that he had French oak. And I said, let me try it.
Yeah.
And it turned out if you have completely the same recipe, same malt, same grain, same yeast, same hops, then if you use French or you use American oak, it's two different beers. If you take them next to each other, you have no question that these are two different beers. In fact, you will not believe me when I say no, but it's the same mechanism. It's just these 4 ounces of oak chips.
That flavor makes all the difference.
That flavor makes all the difference. And the beer made on French oak, you can drink within 3 or 4 weeks of having bottled it. When you do the American oak, it really should best when it has been aging for about 12 weeks before you drink it, because then the whole thing is sort of mellowed up. Beer, when you use bottle conditioning, is a very live product, and I think that at least you can have them around for between 6 and 9 months. By a year, some of them begin to get a little boring.
It's questionable, yeah.
But because they're not pasteurized and you still have their yeast, therefore, doing the little magic at the bottom of the bottle, and that turns out to be the trick.
Well, very cool. Wonderful to hear about one of these hobbies that occupies your time. And now I want to chat about your science, Olaf. Can you tell us sort of the quick and dirty? How do you describe what you do to someone who's not in science?
Well, the short answer is that I want to understand the mechanisms by which all the small molecules that we use to manipulate the function of cells or whole organisms, I mean, human beings, and that do so without any overt evidence that they bind to a target. They just seem to do it by sort of in a non-specific way. How do they work? What is it that they're doing? And we have shown that at least one mechanism is that they go in, sit in the lipid bilayer part of the cell membrane and produce a very specified set of perturbations that are important for regulating membrane protein function, and that we over the years have developed a number of tools that allow us to put numbers to this. And the practical implications of this is that we can both develop better understanding of this concept called polypharmacology, where you want drugs that alter the function of more than one target, as well as we have a mechanism, one at least very general mechanisms by which small molecules are toxic, and we can, in a completely synthetic experiment, we can, with seemingly 100% accuracy, predict whether a small molecule will be toxic or not. So I think that's cool. I really like that.
What a phenomenal description of your work. I love that you tie in those practical implications as well. I think that's so important when you're communicating your science to people outside. And I'd like to hear next about what motivates you. I'm a quote person, so I love getting these little bits of inspiration. So can you share with us today, Olaf, one of your favorite motivational or inspirational quotes?
One of the things you mentioned, that I am director of the Trying to Choose a Link to PhD program, which means I spend a significant amount of my time mentoring and advising budding scientists. So I have a few quotes that I have in PowerPoints. And I think my favorite quote is one by George Bernard Shaw in his Maxim for Revolutionists from 1903. And it is that reasonable people adapt themselves to the world. I slightly changed it. In Shaw's day, of course, it was only men. Mm-hmm. But reasonable people adapt themselves to the world. Unreasonable people persist in trying to adapt the world to themselves. Therefore, all progress depends on unreasonable people.
Very cool.
And the reason I try to instill this into our students is that sort of budding scientists, if they're going to really make their mark, should be iconoclasts. They can't just be sort of the reasonable people who follow the conventional path. They have to be willing to go out and explore something new. They need to have the courage to fail. However, they shouldn't just be completely blue-eyed optimists and be unaware of the fact that there are consequences. So I say there is an Andersen corollary to Shaw's quote, and that in particular applied to people who want to be physician scientists, which is that medicine is a profession with a well-established hierarchy, And you need to understand the system and learn how to maneuver and manipulate. I think the latter is important. The system in order to accomplish your goals. So you have to be somewhat Machiavellian and know how you can go in and accomplish what it is that you want to do.
Well, fantastic quotes. I think those are great examples with applications for science. I mean, I think this idea of being unreasonable, I think you do need to be unreasonable. When you think back 100 years ago, things that they thought were unreasonable then are sort of commonplace now. So we need scientists to be doing the same thing now that in 100 years, people will be looking back and thinking the same thing. So very cool.
If you ever go into a library and sort of just for the fun of it, take an issue out that was one of the bound volumes that were 50 years old, for example, you read and you see people have these vicious debates about concepts that we don't even care about anymore.
Right.
It helps to put everything in perspective.
Absolutely. Well, I think that is a fantastic way to put things in perspective, to get out there, learn the history of science and learn the history of your field and see what the issues were back then and think about how different they are from what's going on now. So very, very cool. Now, when I talk to scientists, I am always curious to hear about who motivates them. Can you tell me about the people in your life and along your career path who really had a big effect on you?
There are a few. I think the most important is probably my first research advisor, Jens Christian Skov, who was professor of physiology in Aarhus, and he was the scientist who discovered the sodium-potassium ATPase, which he eventually received the Nobel Prize for. Skov was really amazing because he had this ability to go with the flow. He was interested in all sorts of things. He could see the humorous aspect of life. He could be very serious when it was necessary. The reason he became a scientist was that he got a rheumatic fever when he was in training to be an orthopedic surgeon. And then he couldn't continue his clinical training, so he went to Aarhus, where he studied the mechanisms of action of local anesthetics. And he did all of that early on lipid monolayers. And then he wanted to do experiments with membrane proteins. And that was what led him to the discovery of the sodium-potassium ATPase. All the while he was doing this, he also was set up the first hemodialysis unit at the University of Aarhus, and his fellows were the people who actually managed the dialysis unit. So he was just one of these people who could identify important problems while having a satisfying career, but he was able to focus a problem with such a laser-like intensity. And I think that sort of his key advice, reflecting his own worldview, was that there may be a few people, say Einstein or Feynman,
right,
whose brain are wired to contribute something really unique. Most of us are smart, we have many different interests, and we really have a rather wide spectrum of careers. I always tell applicants to the MD PhD program that what I'm looking for in a budding physician scientist is really the same as an engineering school is looking for in their recruits. What law schools are looking for, people who are able to think very clearly, who are excited about solving problems, and who are undogmatic about things that come up. You really evaluate in every single situation on its merits. One time I was home at dinner in his house, and then he said, so the real issue is therefore is not to make the right decision, say about a career, because you'll never know whether you made the right decision. You can't do the control experiment, but focus on avoiding the wrong decision. It's not a lot easier, but it changes the nature of the problem, because we may always sort of strive, could there be something better? And there may be something better. This indefinable gut sense still warns us that this is likely to be a bad idea. And so I think that we should avoid what is likely to be a bad idea. And if it turns out we make a decision that was unwise, then be ready to jump ship.
Definitely. Well, I think that is such a great approach. And I think it's hard to have your life planned out 3 steps ahead all the time. So it's great to hear that if you take the strategy, avoid the obvious pitfalls or perhaps, semi-obvious pitfalls and focus on making whatever path you choose the right decision for you, I think is a great approach and wonderful to hear about someone that you've looked up to. And I think these mentors are so important for the development of a scientist. And I want to know when your scientific development began. Can you take us all the way back to the beginning, Olaf, and tell us when you first started getting interested in science and then kind of how you got from there to where you are today.
In some ways, it probably started when I was in high school. I'll sort of take you through my early training because this was in Denmark in the 1960s.
Sure.
So you go straight from high school to university. I was sort of interested in mathematics. I liked physics. I was pretty good at both of them. But there was a problem that my father was a very good mathematician, so that more or less ruled out mathematics.
Right.
But I sort of read voraciously, and I had become interested in the issue of sort of cybernetics control systems, something an American mathematician, Norbert Wiener, had introduced. And that sort of led me to an interest in computer science, but it barely existed in the mid-1960s. And I wrote to the Technical University in Copenhagen, where, because my father told me that they might have that as an experimental discipline, and I got a letter back basically telling me that I was impertinent because first I had to be accepted and then I had to pass my qualifying exam before.
Don't be too presumptuous, yeah.
Don't be too presumptuous. And I sort of realized that if that was their attitude, it might not be a perfect match. But I was interested in the brain, really inspired by Wiener's work. And at that point, I'm not quite sure I can fully defend the decision now, but I decided that it made sense to pursue a medical education. Of course, that would mean you got a really training in organismal neurophysiology. This was close to graduation from high school, and so I decided I better find out whether I can stand being in a hospital or I can handle being with patients. Mm-hmm. And so I worked as a volunteer porter in a small hospital in northern Jutland, where my responsibilities was to move the patients around, to some extent talking to the patients. And I certainly wasn't overworked. I had lots of possibility for surgery. I could participate in rounds. And so I got actually a pretty good insight into what medicine was about. And I discovered that I really liked to interact with patients. I got a kick out of it. So I applied to medical school. And at that time, that meant that I needed to write a letter that should be postmarked no later than I think it was July 31st, that I would show up on September 1 to begin the first day of classes.
Okay.
I think today's medical school applicants are very jealous of that.
I was going to say.
What it meant, of course, was that we were about 300 that showed up, and by the following June 1, there were probably 150 left.
Really?
There was no mentoring, no advising. You were totally left to your own devices.
Sink or swim, yeah.
This was sink or swim, and I think many of us, how we actually proceeded through that training was dependent somewhat on the luck of the small group conference. Most of the teaching was in small group conferences. There were lectures, but say the anatomy professor took about 5 years to go through the body, so I just decided I didn't need to go to any lecture.
Gotcha.
So I happened to be with a group where there were one guy who was somewhat older. He had been a teacher, and he said, I've got to get out in 6.5 years, which was the minimal time you could graduate from medical school at that point, and this was right out of high school. And we talked, and we decided we paced each other well. So I continued this, but I still had this thing with mathematics in the background. So the first 2 years, we had no exams whatsoever. And so the first 2 years, I therefore could actually take about 2/3 of the course load in mathematics. And then after 2 years, I then had to begin studying for what would be a large comprehensive exam where they would test everything we had been learned.
I knew there was going to be a catch to this no exams part, right?
So I stopped the math, and then it was this fantastic experience because you need to do gross anatomy, histology, physiology, biochemistry, etc. They are written and oral exams within a very compressed window. So the only way to accomplish this was that you basically, by early December, had managed to integrate all the different subjects into sort of one coherent understanding of normal human biology. I think that has had a major impact on, sort of say, my outlook, both as an educator, but certainly also as a scientist, because I tend to, even though the research I do is very reductionist, the way I view problems is actually in a very organismal way, very integrated, systems-based way. So I completed the exams, and then I was extremely poor medical student in the sense I almost, except for some of Sko's lectures, I didn't show up to a single lecture, but I was absolutely meticulous in my attendance at the small group sessions so that I would be sure that I stayed on top of all the readings and the material. And I talked to Sko before this comprehensive exam, and he said, why don't I come up and talk to him afterward? And he came up and he said, well, I have this problem. I think it's a really cool problem. It was to take the sodium-potassium ATPase and put it into planar lipid bilayers and then record the currents due to the fact that sodium and potassium would move across the membrane. And at that point, it was known that the pump exchanged 3 sodium for 2 potassium. And that sounded really cool. There was some physics sort of in it, and I didn't know anything about lipid bilayers, but I said I can learn.
Sure.
And there was nobody else in Aarhus. There was nobody in Scandinavia, incidentally, who knew anything about lipid bilayers. So I had a rather rocky start. But one thing that Sko forgot to tell me during this time was that I was not the first person he had suggested doing this project. I think there were 3 or 4 people who had started and left in disgust because they felt it couldn't be done. And nobody, by the way, has done it up to now.
Oh, wow.
I didn't succeed either, but I mean, I was a medical student. I continued not go to any lectures, but I still read voraciously, also in the curriculum. But that gave me a lot of time to just learn how to become a scientist, learn how to become independent. And the way I learned was I read the literature. This was in 1967. I then wrote letters to the people in the United States or in England or Germany whose papers I read and sort of had questions, how do I get to the next point? And they were remarkably generous with their time and wrote back to me and said, okay, now you do this and that. And I finally began to get it to work. I could get membranes that were stable. I could show they had the high resistance that other people who worked with lipid bilayers found they had, but I was unable to do anything with the sodium-potassium ATPase. And then in 1968, I realized that though I really loved the environment in Aarhus, that I wanted to get additional training in biophysics. And at that point, they just had opened a department of biophysics at the University of Copenhagen. A little similar to the situation with the matriculation, it was at that time possible to seamlessly switch between the 2 medical schools. So in July 1 in 1968, I switched my matriculation from being University of Aarhus to be University of Copenhagen and began to set up my own laboratory. It sounds very grandiose.
It does.
At University of Copenhagen. But then that summer I did something that actually was pretty smart. I went to a meeting on membrane biophysics that took place in Jerusalem, and this was just after the 67 war. So there was sort of a special atmosphere there. And after the meeting, I spent a week traveling all over Israel. You could walk through the West Bank without really having the least bit of worry about your physical safety. I went down to Eilat. I went all the way up to Nazareth, and sort of everywhere in between, and had a great time. And I made connections so that fall I would then spend a week at the University of Cambridge and a week at the University of Saarlanders, where I then for the first time could sit down with people who really knew what they were doing. And I was able to learn a lot. That would have taken me probably another year to learn if I had to solve it on my own. But I still, when I look back to this, I can't say I really produced very much, but it gave me freedom. I could work on what I wanted to work on. I had nobody breathing down my neck, nor did I, quite frankly, have an awful lot of advice, but you learn along the way.
Right, you got the good with the bad.
Yeah, and then toward the end of my medical school training, I actually liked clinical medicine, but I didn't have any good role models as physician scientists, whereas had fantastic role models in science. I mentioned Sko beforehand. Jens Otto Witt was an associate professor in the biophysics department in Copenhagen, and he was amazing as a mentor, and he and I talked many times. But the 2 people in Copenhagen who may have been most important for me were actually not faculty. They were the chief electronics engineer, Mr. Peterson, and the one who was in charge of the instrument shop at Deutschland. Because those two guys were willing to listen to me, my ideas about how we should construct new chambers, were willing to sort of take me by the hands, also at appropriate times, sort of smack me down a little.
Right. Tell you what you needed to hear.
Yeah. So that was when I began to really learn how to, once you do single molecule measurements, I think I may have been the third or the fourth person in the globe who actually saw single channel current traces with my own eyes, where I had been completely involved in the design of the whole thing. That was pretty exhilarating.
That is phenomenal.
So I decided early on I would give it a try. And I then spent a year and a half as a postdoc in Copenhagen. And at that point, I'd been an American postdoc at the Biophysics department, Albert Cass. And he said, you need to go to the United States, sort of see an even bigger sandbox. And I went to the Rockefeller University, did a postdoc there. And just as the financing, my fellowship, everything had come into place in Copenhagen, it turns out that my intended postdoctoral advisor at the Rockefeller, his sabbatical at University of Geneva had yelled. So there were two letters crossing each other on the Atlantic saying, I'm ready to come, and the other saying, don't come because I won't be here.
Oh, wow.
But he then decided, okay, I should come. And a former student of Alex Morrow, my intended advisor, Alan Finkelstein, at Einstein College of Medicine, was willing then sort of to take over the mentoring as well as a professor at Rockefeller, Dave Mosrell. And so I actually, therefore, I had a great time. And I find out I very much liked my independence. And so it probably in many ways was good that I could spend most of the time, I was my own boss. I could, a day a week, go up to Einstein. And I spent also a fair amount of time with David Mosrell's lab, and I could do all the various different kinds of experiments I wanted to do, largely without anybody intervening or giving me any kind of trouble about it, nor, of course, did I get an awful lot of advice about how to do them.
Right.
Then I didn't quite know what to do. I mean, I think I really had plans to go back to Denmark, and my girlfriend at the time was still in Denmark. Then I got the offer to move over to Cornell, and it turned out this was in 1973, where there was a major financial cutback in the Danish academic system, and it sort of became clear that why don't I stay in the United States for a few years and ride out the storm? My girlfriend came over, and to some extent the rest is history. Because my chairman of physiology, so I was the first faculty member that he recruited as he became chair, and he is Austrian, and he said I had to agree to stay for at least 3 years. Mm-hmm. And he, of course, knew very well that at that point I had taken the hook and the bait, and there's so much overhead invested in building your own laboratory that when I got a query, would I be interested in a position in Denmark? No, it wasn't quite the right time.
Definitely.
And one day, without ever really thinking about it, it turns out that you have made the decision that I would stay in the United States. Our sons are born in the U.S., and I'm here for the long haul. So at Cornell, I really like the tri-institutional campus with Weill, Cornell, and Rockefeller and the Memorial Sloan Kettering Cancer Center. There are lots of interactions among people, and when you're European and Danish, New York is actually a good city to live in, and we live within walking distance from our work. My wife works at Rockefeller, and so we have actually a pretty privileged existence. We don't need to deal with the immensity of New York City. We live within 10 minutes of our work, and it's been a very good way of living. So for the first 17 years when I was at Cornell, I sort of did well in terms of the academic advancement, etc. And then, but until about 1990, I think I managed to largely fly below the radar in terms of that I wasn't really called much upon to do committee work. But then things changed. A new dean came in, and he was aware of my existence, which I didn't quite know I should be worried about. And so I'd done some fair amount of work actually for the New York Heart Association, chairing the research council for many years. But then I was asked to chair the first conflict advisory panel, or at that point Cornell University Medical College. And that was an amazing experience in terms of how you begin to deal with the multitude of ethical and moral problems that relate to sort of conflicts of commitment, financial conflicts of interest, and so on. And we developed a policy that I'm not sure Caesar's wife would have been able to live up to. And then we had the first case, and everything went from being black and white to shades of gray.
Right. It all looks good on paper, and then...
I think it was fantastic because we sort of began now to say we deal with a lot of situations that have no fully right solutions. There may be wrong solutions in some cases, but how do we come up with something that can let science progress, that protects the investigator? And that's actually something that people often do not really appreciate when they think of conflicts of interest management, that it's just as much protecting the investigator that may be perceived to have a conflict of interest, and therefore it's important that you establish mechanisms that actually to the world as a whole demonstrates there's nothing to be concerned about. So I actually had a pretty good sort of interest there. I liked the intellectual part of it. And in 1995, I became editor of the Journal General Physiology, which I've been associate editor for some years. And that was when I got, I'm not sure it is the best advice I ever got, but Thorsten Wiesel, who was president of Rockefeller at the time, said when I sort of met with him, and I think I got a lot of advance warning. I think we met something like June 26, and I would take over on July 1. But we sat on the terrace outside his office, and he said, so I'm not going to tell you anything about to run the journal. You know how to do that. But there's one thing I want to tell you. Make changes and be sure to make them visible. And when I then sat and realized that I was editor of a journal that at the time was considered to be not quite au courant, how was I going to convince the next generation of scientists to submit manuscript to the journal? Mm-hmm. And so I worked there as a doc for 6 months until so very little happened to the outside world. I was able to reduce the time from submission to first editorial decision by half during this period of time. But outside sort of really low-level mechanics, nothing happened. And then the January 1996 issue was a new journal. And it had exactly the intent that I now realized what it was Thorsten had in mind. I think it just changed the journal immediately at the Biophysical Society meeting in February of 96. There was so much excitement about what the journal had been doing and what it's accomplished. And I think, we really got it to work extremely well. So the other thing you sort of mentioned was the MD-PhD program, and I have been involved in the MD-PhD program for a number of years. And the program became trying to choose in 1991, and there were now 3 programs that ended up being folded into this one triinstitutional program. And there were 3 institutions, each with their sizable egos, that now had to work together. In the beginning, They didn't play very well in that sandbox.
It's no small feat right there, bringing them all together, I'm sure.
I was getting sick and tired of the whole thing in the spring of 1996. I mean, I'd been involved for so many years, longer than anybody else around. So I said June 30th, that sounded like a good day just to say I'd done my part. Then the program sort of ran into deeper and deeper trouble, and same story as before. I was asked whether I would be willing to take the responsibilities for running the program. And the only reason I said yes was that I would have said no if I could have met with the dean. It's one of these situations where when you get a call from the dean's office that the dean would like to come up and talk to you, then you know there's going to be a very big favor being asked.
Absolutely.
So I called my wife and said, guess what? My wife is a very supportive woman, and she said they must be desperate if they ask you. Mm-hmm. Then that evening, we talked about it, and she said, can it be done? And I said, not with the present structure. Can you change the structure? And yes, I said, I probably can. And so I sat down and outlined a new management structure for the program. And much to my surprise, all 3 heads of institutions gave me completely free hands to reorganize the program, which was a lot of work, to take over the program in 2 years of my life, completely. because as editor, I just had to make changes. I didn't really have to break anything down.
Sort of knocking it all over and rebuilding it, yeah.
Completely. That, of course, took a lot of effort, a lot of mental energy, but I must say, I can now look back in 19 years very soon of having done it. Of course, as once in a while, there's things that you look back and say, how did I ever begin?
Why am I doing this? Yeah.
It's been satisfying, and I find that working with the students, advising them, and I'm very firm that I don't solve anybody's problems. I will talk to the students and guide them into how they solve their own problems. And I think they become much stronger for that, and I feel good in the sense that when I let them out into the big world after they graduate, that they are ready to face organizations that are not managed necessarily with the same care, with the same focus on mentoring and advising as we provide. So that's really been a very satisfactory part of my life. I think also in a funny way, it has been good for my science. Because when I took over the program in 1996, this was at a time when this work we are doing on what is the role of the lipid bilayer in regulating membrane protein function, that I had difficulty convincing people that this was important. Mm-hmm. And the NIH study sections did not hesitate at letting me know of their disapproval.
Of course. They're very free with their criticisms.
So I kept the laboratory going because I had a long, productive career studying microphysics of ion permeation. But I increasingly felt that what was really important was to understand how you energetically couple membrane protein conformational changes to the properties of the bilayer, which is something we began to study 25 plus years ago. And I think in the last 10 years, I can say we really have gotten things to fall into place now. We can predict whether small molecules are toxic based on something as reductionist as changes in the granoside channel monomer dimer equilibrium measured using a fluorescence assay in a synthetic bilayer. So we have come a long way. It's been incredibly exciting.
Well, I think that's so exciting to be able to track one of these problems through the years and to see how far you've come and feel like you're finally really digging in there, getting some of those answers. So wonderful to hear about your journey. And I think this tri-institutional program just sounds incredible. So wonderful opportunity there. So Olaf, great to hear about your journey. Now, I'm curious to hear about where you are today. Can you tell us about one of these projects that you're working on right now that you are just the most excited about?
There are two projects that I'm really incredibly excited about right now, and they are almost a little bit orthogonal to one another. So what we found many years ago was that small molecules, surfactants, drugs, phytochemicals can change the conformational preference of membrane proteins, and in particular, the reporter channel that I've used for many years, the granoside channel.
Okay.
And what we found was that we changed the monomer granoside and monomer dimer equilibrium by 2 orders of magnitude.
Oh, wow.
I mean, we saw there just sedate channels opening and closing once in a while, or forming and disappearing, and suddenly it almost went off the oscilloscope screen. It was one of these sort of amazing, I've been privileged to have had them a couple of times, but it's one of these amazing things where you realize everything you thought about a certain part of the world or science or mechanisms was just wrong. I was incredibly excited by this. Though I was at that point still mostly focusing on the microphysics of ion permeation, I knew this had to be important. So we began to explore this, and when I then applied for competitive renewal of my grant, the study section was thoroughly unimpressed by my excitement. And so...
Really? That's devastating.
Well, I had another grant about a problem that I wasn't that excited about. So I just decided I would cannibalize that grant and make sure that I could move this membrane project forward. So we did that, and we developed the theoretical tools that we actually could not just say we have a membrane effect. I think what I really have contributed to this field is being able to assign numbers to the observations, because it's not just that something goes up or something goes down. It actually matters a lot whether it goes up by a factor of 3 or a factor of 10 to the third, or even a factor of 10. Then we began to deal with a question of really biologically important small molecules, the molecules that you use to manipulate all sorts of biological functions. And what we showed was that at high concentrations, platelet activating factor becomes a very potent modifier of bilayer properties. So the first thing we really did, I would say, that put us on a new track was a molecule called genistin. And it turns out that it changes function. And that led me to a new, different line of research, because it turns out that almost all of these biologically active phytochemicals, what you see in the health food stores and so on, rather potent modifiers of bilayer properties, which almost certainly is not why they're good for you when you eat them. I would say one should stay far away from the concentration where they change bilayer properties. But we have used this to show that a lot of the results that people publish, these effects occur at concentrations where we can tell you that it is a very potent modifier of the elasticity of lipid bilayers.
Gotcha. So it's not something you would ever encounter sort of in food, in nature, those sorts of things?
If you got up to those concentrations, you would have other problems that you had to deal with. You would deal with cardiac arrhythmias and everything else.
Gotcha.
So we took this partly because a lot of what we have been doing had been in using single molecule detection methods. So we needed to have a method that would allow us to detect whether molecules, and preferably libraries of molecules where you to some extent change only a few parameters one at a time, how they change the physical behavior of the lipid bilayer. And to do so, a student, Helge Ingolfsen, and I developed a fluorescence quench method. And now we can use changes in quench rate as a measure of the changes in the average number of channels in the vesicle membrane. And now we're talking business.
Yeah, it's very clever. I like it.
Now we might take a week to a month to characterize a molecule by single channel method. We can do 10 a day. And one student was able to do 60 experiments in a day, but he had to almost be carried out of the lab.
Wow. It was a full 24 hours of work, right?
So we did this, but still there was something missing. And about a year and a half ago, I then had lunch with the director of the high-throughput screening facility at the Rockefeller University. One of a student in my lab sort of developed a contact because she had been talking to him about some chemoinformatic issues. And we were talking, I told him about what we were doing, and he sort of said what they were up to. And then he said, but I have this frustrating situation that we have characterized a large fraction of the Rockefeller chemical library where they provide compounds to investigators at Rockefeller of different. And we have characterized them in terms of their toxicity. Most of them are not toxic, some are toxic, and some are very toxic. And they used very sophisticated toxicity assays. So it's not just that you have to blow up the cells, apoptosis markers, nuclear morphology markers, etc. And so I said, okay, this is exactly what I'd be looking for, because now I could get a blinded library. And I said, just give us 20 non-toxic, 20 moderately toxic, and 20 toxic compounds, and we will see what are their bilayer modifying properties, and if the changes in toxicity in any way tracks with their bilayer modifying potency. And what we found was, much to his and my amaze, I thought we might find it, but I didn't know that the results would be so clear, that every single molecule that changes a fluorescence quench rate by more than a factor of 3 is toxic. Actually, every molecule that changes more than a factor of 2.8 is toxic.
Wow.
Of course, molecules can be toxic for all sorts of ways that have nothing to do with the membrane. And so even if they are basically have no effect on the membrane, about a third of them are toxic, which probably in the way this particular library was chosen, that's what it is. But so the point is that now we have a sort of physical chemical measure that people can come in at 9 a.m. in the morning, and by basically before noon, I can tell them whether this molecule is likely to be worthwhile pursuing in terms of future studies, or whether they should just forget about it. If it does more than threefold, I would say forget about it. It's likely to be too toxic for any useful stuff. If it is very little effect, well, it could still be toxic for all sorts of things we don't think about. And in the higher bilayer modifying effects, I'll come back to that in a moment, we just don't know. But if you were selecting molecules that you were going to invest a lot of money in future drug development, I think I would tend to put my eggs in the basket where you had the molecules.
Right.
But now imagine that you're the medicinal chemist and you have worked hard to make this library, and then I come and tell you, forget about it.
You've lost a friend at that point.
Well, yes, unless you can say, but can we build a better mousetrap? So this morning, for example, I actually met with some colleagues here at Weill Cornell who are using a specific family of phytochemicals to develop treatments for leukemia. And they find these molecules are basically inert or very little effect on normal cells, but really sort of have a much higher lethality for leukemia cells. And they knew some of them are likely to be toxic, so they made a variety of analogs of these molecules. And just looking at the structure and seeing where the various moieties that they had put on the molecules, you could sort of, without doing anything sophisticated, well, you can rule out mechanisms. And you could sort of begin to say, where would you be able to modify the structure in such a way that you would minimize the undesired effects, but still maintain the desired effects? And then it becomes important that you have a mechanism. It doesn't help you that you give the molecule to the safety people and they tell you it's toxic.
Right.
Then you're back to square one. But if they say it's toxic, but see, it's also very bilayer modifying, and you look at the structure and you can say you more or less can predict how it's going to sit in the interface. And then you can say, if you go in and you make changes in this part of the molecule, it would be worthwhile to re-examine whether it hits the target that you have in mind and that you have now reduced the possibility for deleterious effects. And that way you have your friend back.
This is true. You won him back. That was like a heart-wrenching roller coaster moment there. Lost a friend, won him back.
Yeah. So then we have a molecule you may have heard of, amiodarone, which is used to treat atrial arrhythmias.
Okay, yeah.
It is very hydrophobic. It changes the function of numerous membrane proteins. Nobody really knows how it works. And a former student of mine who is a board-certified cardiologist suggested that we explore what amiodarone does. And an instructor in the lab, Rada Rusanova, decided this was a cool project. So we have now shown that amiodarone at the concentrations that are clinically relevant alters lipid bilayer properties. And that comes back to something I mentioned, I think it briefly very early in the conversation, that now we have a mechanism for polypharmacology, because molecules that change the physical properties of the bilayer will change the function of all the embedded proteins. Changes in protein function will not be the same for every protein because it's the difference in bilayer deformation energy for the two conformations, which may vary widely. So we can demonstrate. We have done that. We have developed test cases to show that we can, in a system that has no chemical specificity whatsoever, that we can actually have functional specificity, seemingly functional specificity. So we are now exploring whether we actually have found the mechanism by which amiodarone works, or does some of what it does, and which would sort of be cool because it opens up for new class of pharmacological approaches to treat difficult to treat diseases. I mean, when you treat a system disease or circuit disease, do you want to only hit one target, 100% of the effect on one target? Or would you prefer to hit 10 targets with 10% of the effect on each target? I haven't really done this in a quantitative manner, but it seems to be a little more stable if you sort of spread the load over the system and therefore are not completely dependent on just the properties of one target. So those are the things I'm really excited about right now.
Oh, that is absolutely fascinating. It's been wonderful to hear about your work and really dive into some of the details of these lipid bilayers. I think it's something we all take for granted on a daily basis, but like you're describing here, there's so many applications for pharmacology for all different areas of research. So very, very cool. And I think you touched on a lot of awesome examples of the ups and downs of science. I mean, I think you have these super exciting, thrilling moments where you're making a discovery, where you're one of the first people to see this phenomenon with your own eyes, but then also sort of the challenges and the struggles that you have to work through to get things to work. So wonderful to hear your story. And we chatted a little bit in the beginning of our interview, and you mentioned you like to read. And I myself am an avid reader as well. And I love to get book recommendations from people. So Olaf, can you tell us about a book that you've read and really enjoyed?
One book that I think should be read by every scientist and probably many lay people, and I mean, it was just made into a PBS miniseries, is Siddhartha Mukherjee's Emperor of All Maladies, a biography of cancer. Mm-hmm. It's very well written. He writes incredibly well. But it also is a really fantastic analysis of sort of really the progress of research, sort of the relationship between the advances that occur in the laboratory and then the advances that usually much later occurs in the clinic. And how in cancer, really many of the advances that took place were not necessarily tightly coupled to advances in the laboratory. And I think so both to understand better cancer as a disease, which I think he does remarkably well. I think it also is worth reading because it provides a lot of insight into what should we expect when we say we need more financing of research. What can we reasonably then tell the U.S. taxpayer who ends up footing the bill? That's what I really like this book, because I think he manages really to get the excitement across, but also sort of, wait a minute, it's not going to be necessarily be all that simple. Mm-hmm. It's important not just to tell people about all the excitement, but also tell people about all the uncertainties and the likelihood that there's going to be mistakes made along the way, just because we, by definition, work in areas where we can't look in the back of the book and see what's the answer to the problem.
Right. We're dabbling in the unknown.
You're dabbling in the unknown. And I think that book does it remarkably well. So that's why I think it is such a great book. Right now I'm reading Daniel Kahneman's Thinking Fast and Slow, which I'm really fascinated by. I'm not that far into it yet, but one of my colleagues recommended it to me, and I really think it tells us a lot about how we make decisions and how we can be fooled into thinking we make valid decisions, although we are completely just winging it. So that'll sort of be on my bedside for a little while, because I find that it's an incredibly important book that I think almost anybody who is involved in decision-making, advising, but also I think in science can learn a lot from that book.
Well, great recommendations, Olaf. I will put them out there on our website for our listeners to find. And we chatted a little bit earlier in the interview about one of your opportunities to travel. You mentioned you went to Jerusalem fairly early in your career, and I love to hear about these travel experiences. I know this is one of the great parts of the career. So can you think back now on all the places that you've been for science and tell us about your favorite?
There's one. It was 1991, and I was in Bangalore in India. And the reason it was such a special experience was that it was just before the first Gulf War. In fact, the Gulf War broke out while I was in India. So this was one of these U.S.-India meetings that were financed. Basically, the Indian government was using rupees to support these meetings, and it was repayment for various funds that United States at earlier time had given to India. It was a meeting on membrane transport, lipid bilayer structure, and the science was great, but there were some discussions at home before I went. Could you go? Because what was going to happen in Iraq and the Gulf? Mm-hmm. The organizers were friends of mine, so we decided, no, I should go. And as we went there, a good friend of mine said, oh, I have some bad news for you, because he said, my neighbor said, don't worry, nothing will happen until I'm called up. And he said about 3 days before we were sitting in the plane going to New Delhi, this guy had been called up.
Oh, no.
So we said, well, that's interesting.
Right. Tensions are rising.
Some people came as far as Frankfurt and turned around. And some people came there and said, this is getting too hot for us. So they went back before the meeting was over. But there were a hearty core of us. And because our Indian hosts had done so much to make this a great meeting, I think about half the attendees had decided not to show up. Mm-hmm. So they obviously understood very well why people did not show up, but everything that they had worked so hard for seemed to some extent be wasted effort. So those of us who were there were treated like royalty, because they had the time to do it, and I think they appreciated the fact that we did come. And in terms of environment, being able to talk to the students in India who are very smart, I think it is the best meeting I've ever been to. It was certainly lots of ideas. I recruited postdocs who later came to my laboratory from that meeting. I developed many friendships from that meeting. And I will also say, we actually realized when the war would start, because we were sitting there every morning reading the newspapers. Was it going to just become back to normal? Was this going to end back and down in downtown Bangalore? You saw these billboards where you saw the angry Iraqi camel and the American eagle sort of confront each other there. So you sort of felt this was beginning to be a little iffy.
Right.
And then we realized, okay, we knew exactly when the war would start. Everybody said it was going to be the fact that Dan Rather could talk about it at the news in New York. No, it would start so that the jets could bomb Baghdad at night, but land on the carriers in light. And then we sort of back-calculated roughly where would the carriers need to be, how much time, and then we were within half an hour of when the bombing actually began.
That's impressive calculations. Yeah.
So we were sitting there, the latter part of the meeting was in Hyderabad, and I still remember we were sitting that morning, and our host came down looking very somber and said the war has started. Then the TV came on, and a red turbaned sheikh, or Air Force general in India, said that the United States would just get their rear ends whipped. And so that made us feel very good. And while we were there, Pan Am had gone bankrupt. Mm-hmm. So that was a carrier that took us out there. And so we sort of were...
Couldn't catch a break. Yeah.
We were a little nervous. I called regularly my wife, and she said, if nothing else, you can always go back over the Pacific. And so the real question at that point actually would be, if there were a lot of casualties in the war, then all the planes would be used to transfer casualties out of the field into Europe and to the United States. We took off basically at the designated time. And then we, of course, wanted to avoid the Iraqi airspace, which meant, I don't know if you remember at the time, there were a fair amount of unrest in the Baltic states. And so we, of course, said, I mean, now just going from, sort of say, the fire into the frying pan. I mean, this just seemed a little too much that we went this far north, but at least we came home, not a problem. And I still think that I was very happy I went to this meeting. I think I got an appreciation of India and as a state and Indian people as a community that I would never have gotten any other way. And I've been in India a number of times since then, and it's the first meeting that really stands out in my mind as a fantastic experience.
Certainly. And just the idea of being there, being overseas for a piece of history and really seeing it from another culture's perspective, I think is really something you can't get every day. So very interesting to hear your story and to hear about this travel experience and kind of all the ups and downs of it as well. Going back to that roller coaster ride, I think you should write a book, Olaf. And I think you brought up a really great point in the story in talking about the people, how the people for the conference, they'd worked so hard for it. They were gracious hosts. They were sort of showering you with attention because they could. And I think in science, you come across these just very generous, genuine, and wonderful people. And that's one of the things that we love to showcase on our program, to kind of talk about the human side of science. And I think when you look back across all the groups that you've been a part of over the course of your career, there's a lot of traditions that people remember, or even just funny or fond memories. So can you share one with us today, Olaf?
So I think we are all, to some extent, foodies. And so whenever anything happens, we always go out and eat lunch together. The Rockefeller Faculty Club is a good place where we go to celebrate whenever a manuscript has been accepted. We don't go out and do things such as having, say, movie nights or anything like that, but we like to have people home to our apartment and really sort of pamper them to let them know how much we appreciate what we are doing together. And so I would say it's more a little bit family style, our interactions. But in science, you have people who are very smart, who are usually quite opinionated. And so if you just have 2 or 3 people in the lab, you're sure to find that any topic you ever bring up is going to be subject to intense discussion with quite diverging views. And what I try to do is actually to promote these discussions, that we all get a little bit see that there are many different ways that you can view the world, that you can approach the problems from. So I think we are probably a little boring. We are not extremely exuberant, but we have a really good environment in the lab where we are able to see all the little idiosyncrasies of life and smile and sometimes laugh and sometimes, of course, swear at them. And so it's a lot of fun.
Absolutely. I think you laugh and you cry together as a lab. I love this description of a family style interaction. I think that's a great way to describe it because you work so closely together. People are very passionate about what they're working on. And a lot of people are coming in with different perspectives. Everyone's sort of looking at an issue a different way. So it's great to get together outside of the lab, talk about things that are maybe outside of science and just learn about everyone's perspectives. So.
Oh, yes. Those we talk a lot about, say, books that we read and ideas and so on. Yeah.
Absolutely. Well, while we're on the subject of big ideas and talking about things that fascinate us, I want to hear about one of these big ideas that you have, Olaf. What is one thing that you would love to answer but haven't had a chance, maybe because things like funding, staff, technology, feasibility, any other obstacle is standing in your way?
So it sort of is a logical continuation of what I told you about, but I think we now have managed to make this idea that small molecules can regulate biological function by interacting with the biolayer. I think we have made that sort of appropriate for conversation in mixed company. And we have some insight into the underlying mechanisms, largely based on our experiments in synthetic systems. If you look carefully at the papers we have published, you can see that there are some really big question marks hidden in the results we have published. And these are all very hardcore biophysics. So it's not so obvious to me that one could write a grant that would be funded to do these studies. But if you said that funding is not an issue. So if I really was going to do that, I would say I would take these things that we are doing in the synthetic systems, and then I would extend them to, say, people say, well, you have to study lipid compositions that are similar to those in cell membranes. Well, that's not possible. Cell membranes are compositional heterogeneous in the plane of the membrane and across transverse to the membrane. We have no idea really how the membrane proteins interact with the membrane lipids to create an environment that is completely different from what you would get even if you take the membrane lipids and make an asymmetric biolayer. So what I would like to do, and we have some hints that we may be able to do so, is to using the fact that we actually in some cases really know what we are doing, and then take these approaches and go in and pursue them in cell membranes with the same, so to say, ferocity with which we have done it in the synthetic systems. And say, do something which would be even, you just change expression levels of membrane proteins in the cell membrane and begin to see, does that actually couple to changes in biolayer properties that we can record using our various probes. Say, if you load them up with voltage-dependent sodium channels, cells, would you then find that there would be voltage-dependent changes in the function of a reporter channel, reflecting the fact that the sodium channels change shape as you go between a closed and an open inactivated state. So these are the kind of things where I think we are probing on questions that are rather fundamental to understanding how all the functions in a cell membrane are integrated, and also become very important for how you will pharmacologically modify the function of these proteins, because a lot of the druggable targets in our body are membrane proteins. Mm-hmm. And so if we could come in and get a better understanding of all of these things and not treat membrane protein just like a glorified globular protein, by which I mean that you really forget about all the constraints the bilayer imposes on the structure and therefore the function of the membrane protein, that would be incredibly exciting. That's something I would like to do. And then there is, for example, malignant cells have different membranes than normal cells. Of course, in terms of the proteins and the receptors, but also there are some hints in terms of the lipids. And if we actually knew how to exploit that, then we would have completely new tools with which we could begin to approach the treatment of malignant diseases, which I think would be incredibly exciting to contribute to.
Absolutely. Well, outstanding questions and approaches, things in your mind that you would love to see happen in science. And I think every scientist has a list, probably a really long list, of these questions that they would love to answer. So it's fun to dream big and talk about them. And it's been wonderful to chat with you, Olaf. I love to end our interviews, though, with one last piece of advice or inspiration. So do you have a little pearl of wisdom that you could share with myself and our listeners?
Yes, it's one I very often tell to students. Always distinguish carefully between what is interesting and what is important. You work on problems because you think they're interesting. You get other people to listen to you, to read your papers, to fund your studies, because you are able to convince them that the problems you work on are important. I don't care, quite frankly, if somebody comes and tells me I'm really interested in this problem, in one particular problem. Well, that's very nice. But unless they can convince me that it is important, and I mean, of course, what I think is important, you may find to be completely irrelevant, and vice versa.
Right.
But unless people in their own mind always say, if it's worth my spending time on it, it has to be important. It's a small thing, but it really is very helpful. It is invaluable when you're writing grants, when you're, to some extent, when you're writing manuscripts, because the politicians may not be willing to give us money. But I think the taxpayer overall is supportive of high medical research. But it's because they think or they're convinced that we work on important problems that have real impact on health. First of all, the preservation of health, and when that fails, treatment of disease. So, as I said, always distinguish between interesting and important, and it's the latter that is important.
Well, fantastic advice. Listeners, keep that Venn diagram in your mind of your interest and the importance, and always remember that when you're thinking about science questions. Now, Olaf, fantastic conversations we've had today. Can you tell our listeners how they can learn more about you and your work?
My email address is Sparre, SPARRE, @met.cornell.edu. I'm happy to receive emails. You may need to be patient. I get a lot of emails every day, but if anybody wants to discuss these things with me, I would be very happy to do so.
Fantastic. Well, we appreciate it. Olaf, thank you so much for joining us today.
Yeah, and thank you very much for inviting me to be here.
Absolutely. It was a delight. And thank you, listeners out there. We'll see you next time on another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.